Dear students, welcome to Tumala online classes. In the last video, we had started with one topic, that is prophase 1 in meiosis 1, right? In that, 5 stages that we come across: leptotene, zygotene, packetine, diplotene, dichromasis. In that one, leptotene I had finished in the last class. Now we shall start with the zygotene now. Next stage is zygotene. Zygotene is also called as zygonema. Zygonema. In this stage, what happens with the chromosomes is very important one. Here the chromosomes become little thicker. I shall show the chromosomes over here in the zygonema stage. What happens over here with the chromosomes is the chromosomes, that is homologous chromosomes. I have taken a pair of homologous chromosomes over here. They are very thin there, and these homologous chromosomes, what happens with themselves is, see their reversal. So I will draw this diagram like this. So this is one chromosome, and this is the chromosomes with 2 chromatids. This is also another chromosome. So both are similar in their structure, hence homologous chromosomes. These are called as, okay. So one is shaded chromosome. I am going to show over here. One is shaded one. This is one chromosome. For knowing what kind of differences that we come across in these stages, so as to know that one, I would like to show, I would like to shade this chromosome now, right? So these are 2 homologous chromosomes which are similar in their structure, morphology, etc. Okay, these chromosomes in zygonema stage, so both of themselves overlap on each other, right? So when they overlap on each other, how do they look like? See here, this is the centromere of one chromosome, this is the centromere of another chromosome. See here, this is one chromatid of one chromosome. See here, these are the chromatids of one chromosome, and another chromatid of another chromosome. See their reversal, you are observing here. So both of themselves are overlapping on each other. So they are attaching each other. They lie on each other. So these chromatids of one chromosome, one homologous chromosome, are called as sister chromatids. Remember, sister chromatids. These are the sister chromatids of this homologous chromosome. These are the sister chromatids of another chromosome, homologous chromosome. So one is brought from paternal set, and another one is brought from maternal set. Okay, paternal in the sense that father, maternal in the sense that mother. One chromosome is from mother, one chromosome is from father. So both they overlap on each other. The process of this overlapping of these chromosomes on each other is called as synapses. Right? What it is known as synapses occurs over here in these chromosomes. So one of the chromosomes is shaded over here. Remember, how to shade this chromosome. Okay, this is a shaded chromosome. Actually, there is no shade. For understanding purpose, I am shading this one. See here, this is one chromosome. This is another chromosome. Now you can understand what is synapses. And these chromosomes actually within the chromatids, so they attach somewhere else. In some points they attach each other. So the way of attachment is of three kinds. See here. This synapses that occurs in three ways. One is called as procentric synapses, and the second one is called as pro-terminal synapses, and third one is called as random synapses. How synapses occurs is procentric. Pro means first. First the overlapping starts from the centromere. This is what? Centromere. If the synapses are overlapping starts from the centromere side, then it is said to be as procentric synapses. Means attachment initially takes place near the centromere, and later the gradual attachment that occurs over here. And pro-terminal. Pro means first, terminal means tip. If the synapses or the attachment starts from the tip of the chromosome. Say for example, if this is the chromatid of one homologous chromosome, this is the chromatid of another homologous chromosome. So this is the centromere you consider. This is the centromere. From the centromere the attachment starts, then it is said to be as what? Procentric. If the attachment starts from the tip gradually like this, then it is said to be as pro-terminal. And random means the attachment of these homologous chromosomes take place so randomly on certain points. On certain points, then it is said to be as random. These are the three methods of synapses that we come across over here. So any one of the type of synapses may occur over here, so that these chromosomes are attached at certain points. This is one point, this is another point, this is another point, like this. So on certain points they may attach over here, and that point, how it looks like is ladder-like. The attachment itself is just like zipper-like. Zipper-like you can understand. The attachment slowly, gradually takes place like this. So that way it is called as zipper-like, and they are attached at a certain point which looks to be as a ladder-like. So that kind of point that we come across in between the two homologous chromosomes. I shall draw that diagram, related diagram. This is one chromatid of one chromosome, and this is another chromatid of another chromosome. See here, at this point, a ladder-like attachment that you come across over here, and that is that ladder-like structure. Synaptonemal complex it is called as. What it is known as? Synaptonemal complex. Right? Synaptonemal complex it is called as. That kind of synaptonemal complex is formed between two homologous chromosomes at certain points where they attach each other. Right? So it's very clear, I hope now. As a result of which the same genetical aspects have been joined in homologous chromosomes, right? So that is very important point to be noted. And whenever the chromosomes attach each other, and these chromosomes are totally called as the bivalents. What is the called as bivalents? Bivalents, whenever you see the bivalents, two homologous chromosomes, one is from paternal set, one is from maternal set, which are similar in their structure, they overlap on each other. The process is called as synapsis, and the synapsis takes place in the three ways. Any one of the way it takes place, and after synapsis the chromosomes are said to be as bivalents, and these are also said to be as tetrads. These are also known as tetrads. Tetrads in the sense that there are totally 4 chromatids that we come across now, right? So there are 4 chromatids that we come across over here. Okay. So this is how the zygolemma stage in which the chromosome arrangement that take place. And the next one is packaging. In packaging, what happens is, see here, if this is the cell, the gradual movement of the centrioles is taking place, of course, here, right? The gradual movement of the spindle fiber is going on, of course, from each ester. So it is quite natural, and this is the nucleus, inside which a pair of homologous chromosomes I have taken over here. So here in this case, after synapsis, what happens over here is, so in this case, this is one chromosome, okay. After synapsis, the extent of the genetical material that takes place over here. What happens over here is, this is the shaded chromosome. The point at which both the chromatids, non-sister chromatids of 2 homologous chromosomes. So this is the shaded one. Yes. See here, whenever they attach at a certain point where the ladder-like protein complex is formed, and that ladder-like protein complex, which is said as the point of the synapsis. So there after that one, these non-sister chromatids. What are non-sister chromatids? You may ask over here. See here, this is one chromosome, this is another chromosome. The chromatids of this one and this one are non-sister chromatids. Okay, this one as well as this one are sister chromatids. So these non-sister chromatids overlap on each other or cross over each other like this. When they cross over like this, in between these two non-sister chromatids, the point where they attach each other, at that point there we come across recombinase nodule. Recombination nodule is seen over there. If this is the chromosome, what we can take over here. So they overlap on each other. So here at this point you will come across a nodular structure, and that is called as recombination nodule. Recombination nodule at that point where they attach on each other, right? So and this recombination nodule mainly consisting of enzymatic complex. It consisting of an enzymatic complex, and that enzymatic complex mainly consisting of endonuclease enzyme is present over there. Endonuclease is there. Ligase is another enzyme is there. So like this, there are different types of enzymes are present in the recombination nodule itself. In the recombination nodule. When these two non-sister chromatids overlap on each other in between that point, so recombination nodule is formed. The recombination nodule is containing enzymatic complex, and this enzymatic complex mainly consisting of endonuclease is one enzyme. And this endonuclease enzyme that cuts the point where both the chromatids, non-sister chromatids overlap on each other. See here, consider this one is a shaded one. This is shaded chromosome. For your understanding purpose, I am going to share this one. Okay. Okay, like this. See, so at this point, you will find this one. Okay, like this. Here, the cutting of this chromatid here and the cutting of this chromatid here that take place. Both of themselves cut together. This chromatid that cuts, this chromatid that cuts. So, and the exchange of these two chromatids that take place over here. Exchange of these two chromatids that take place over here. Here also it is the same. If it is lying like this. You can observe this. It cuts over here, this cuts over here. So that what happened over here is the exchange of these two chromatids that take place. When the exchange of these two chromatid pieces that take place, the genetical recombination. Why? Because here are many number of genes are arranged over here. This is combination of gene what we call it as. And at the tip there is a combination of genes, and that combination of gene comes to this chromosome, and this combination of gene comes to this chromosome, so that recombination of the genes that take place over here between two homologous chromosomes. And that process, the exchange of the genetical material between the two homologous chromosomes or the non-sister chromatids is called as crossing over. It is called as crossing over. What it is known as crossing over. The exchange of the chromatids or the exchange of the genetical material between two homologous chromosomes, right? Or the exchange of the chromatids between two homologous chromosomes is called as what? Crossing over. Means the major aspect what takes place in parenting is crossing over. The major aspect what happens in case of this zygote is synapses. The major aspect what happens in lambdotin is bouquet stage, it is called as. So these are the unique features that you must remember in each step over here. So this is about what? Packaging. Packaging. Crossing over that take place in the two homologous chromosomes, so that genetical recombination occurred in pachytic stage. Genetical recombination. Genetical recombination that takes place over here in this stage. Next stage is, next one is diploidy. In diploidy stage, what happens over here is, in this stage, usually the two homologous chromosomes, what we come across here is, this is one homologous chromosome, this is another homologous chromosome. Okay, so this is now the exchange of chromatids that take place over here. What happens? This is like this, and this one is like this. The shaded one comes over here, unshaded one goes over here. So actually this is the shaded chromosome. So like this. The shaded one went over here. Exchange of the chromatids that took place at the point where both of themselves attach. See here, the exchange of the chromatids that take place, and this process itself is called as crossing over. So that takes place in case of packaging. In diploidy, what happens over here is, these two chromosomes move apart. When these two chromosomes move apart, so how it looks like you can see over here. This is one chromatid, this is another chromatid. So this is one chromosome and this is another chromosome. See here. How it looks like? It looks like X mark. It looks like X, and this point is called as chiasmata. The X-shaped structure itself is called as chiasmata. The chiasmata is seen in case of this diploidy stage whenever both the chromosomes they move apart. Sometimes the same structure, so if they move apart a little wider, the moment it looks like O shape. So it may be X shape or it may be O shape. Sometimes it looks to be as, and this X shaped structure is easily seen and it is called as chiasmata. So the chiasmata formation is seen in between two homologous chromosomes, so that you can observe over here. So this is one homologous chromosome which takes the shaded part of the chromosome. This is another chromosome which is shaded one. Okay. It looks like X-shaped, and the X-shaped structure formed where both homologous chromosomes meet each other. The X-shaped structure itself is called as chiasmata. The chiasmata formation that takes place in diploidy stage. This diplotin stage has its own importance. What is the own importance of this diplotin stage? In human females, it arrests from the 5th month of embryo up to 12th year, almost 12th year of the young one. Up to this year, this many years, it is arrested. It is arrested up to the puberty, what we call it as 12th year, in the sense that up to puberty. Means the female get maturity here. That maturity itself is called as puberty. Up to puberty, so what happens? It arrests. In this stage only, it arrests in the oocyte itself. In the oocyte formation, so during the cell division, so meiotic division that takes place during what? During the formation of gametes. Oocyte is going to form what? Ovum or egg cell. Oocyte is going to form ovum or egg cell. During that period, so it rests in the 5th month of the embryo up to the 12th year of N1. So that is up to puberty, or it is called as ovulation. Ovulation is nothing but the process of release of ovum or egg cell, right? Release of the oocyte. So from the ovary is called as ovulation. The process of releasing of the egg cell or the oocyte from the ovary. So ovary is called as what? The ovulation. Up to ovulation or puberty, so what happens? It arrests. Arrest means floating stage. It arrests in human female. Floating stage. Arrest up to 12 years in case of females. And this is the stage where the giant chromosomes like Lamb-Brush chromosomes are formed. Giant chromosomes or Lamb-Brush chromosomes are formed in the oocyte of some of the lower organisms like fish, amphibians, right, as well as reptiles, sometimes birds here. So there the Lamb-Brush chromosomes or the giant chromosomes are usually found over there, right? So this is about the diplotin stage, and the last stage is dichromasis. Last stage is dichromasis. In dichromasis, what happens? So basically, in diplotin stage, the chromosomes move apart, and that movement move apart of both the homologous chromosome and looks to be as X-shaped or ultimately O-shaped. So, and this repulsion, what it is called as repulsion process, it is called as. Both the homologous chromosomes repulse each other. When the repulsion continues here in dichromasis, chromosomes become very thick and sharp in structure, and the repulsion of two homologous chromosomes continues, and ultimately they move apart each other. They move apart each other. Means, so this charismata what is formed over here, gradually what happens over here is, it moves, it slides out. When it slides out, both the chromatids, non-sister chromatids, separate from both the homologous chromosomes. So that what happens over here, here one chromosome is, and this is another chromosome. This is another chromosome where the charismata is seen over here. And gradual movement of both the chromatids, this moves like this and this moves like this. So when these two, this moves like this and this one moves like this. When these two separate each other, so taking out their respective chromatids, right? So when they slowly, gradually slide out, so that process itself is called as a terminalization. Terminalization. Slowly, gradually, these two chromatids slowly slide themselves and ultimately they separate themselves. Right? And now, independently, how the chromosomes are looking like. I shall show you here. So, this is one homologous chromosome. This is another homologous chromosome. So, this chromosome is having see here, non-shaded chromatid is present over here as a result of the crossing over. Here also the same. See here also. Now, what about this one? It looks like this. Looks like this. This is one homologous chromosome, this is another homologous chromosome. After terminalization. Terminalization means what? The repulsion of both the homologous chromosomes that take place gradually and ultimately both of themselves separate. Right? Now you are observing here the combination of the genes. This is one gene, like this. The combination of the genes that change. Why? Because the combination of genes in this region of this chromatid is different, and this comes over here, the combination of genes that changes. It is called as genetical recombination. It is called as, right? What it is known as genetical recombination that usually take place in packaging stage during crossing over. After crossing over, charismata formation is seen in case of diplotin, and in dichromasis terminalization process. See, in each step there is one unique word is there. In leptotin, bouquet stage, zygotin, there we come across one more. What is that? Synopsis. In packaging, crossing over. In diplotin, it is what? Diplotin stage. So where the crossing over that completes there, charismata formation that takes place, and in dichromasis, terminalization takes place. Ultimately, the chromosomes are just like this only. Okay, so this is how where the arrangement of chromosomes and the genetical material that exchange that take place in between the two homologous chromosomes. Like this, I have shown over here only two homologous chromosomes in the diagram, but actually there 23 set of chromosomes will be there in case of human being. So remember, all are doing the same type of changes, means so they are bringing about some changes within themselves. Similarly, as I said, taking an example of two homologous chromosomes over here. Okay, so these are the 5 stages that we come across in prophase 1 of meiosis 1. In meiosis 1, prophase 1 has 5 stages. For our understanding purpose, we have divided prophase 1 into 5 stages there. Leptotene, zygotene, pachytene, diplotene, dichromasis. Like this, 5 steps. Each step is very important for NEET examination. In each step, so there occurs one important feature, that is bouquet stage in leptotene. In zygotene, there we come across synopsis, and in pachytene crossing over occurs and genetical recombination of the genes that take place. And in case of diplotene, there occurs, so in oocyte usually it is arrested for about a longer period up to the 12th year, up to the puberty of the human female. So there it is very important aspect to be remembered there, right? So and one more thing in case of diplotene is, so what happens after crossing over? So the chiasmata formation that occurs over there, and ultimately in last step, that is dichromasis, terminalization that occurs over here. 2 homologous chromosomes, so which have shown the crossing over. After crossing over, the sliding of both the chromatids, non-sister chromatids that take place over there, and as a result of which, so they gradually slide out, and that is called as terminalization, which take place in the last stage, that is dichromasis. Along with this one, in dichromasis, one more thing to be remembered, that is nothing but the gradual disintegration of the nuclear membrane take place. When the disintegration of the nuclear membrane take place, disappearance of nucleolus also take place. Disappearance of nucleolus also take place. And that shift to the next stage, that is metaphase 1. What actually happens in metaphase 1? Let us now study it. So almost all the chromosomes, that is homologous chromosomes, they lie directly into the cytoplasm. Now imagine yourself, all the chromosomes are in cytoplasm directly. Why? Because the disintegration of the nuclear membrane has taken place and disappearance of nucleolus has taken place, and both the esters are on the respective poles and the spindle fibers are completely formed there, right? So now in metaphase 1, what happens? Let us see. It is also similar to the mitotic cell division itself, so but a little bit change that we come across here. So what is that change? So let us study now. Metaphase 1. Metaphase 1, what happens over here is, if this is the animal cell, then disintegration of the nuclear membrane has taken place. There is no nuclear membrane is seen and nucleolus is seen. Only the chromosomes are lying over here, and both the chromosomes I am showing over here. So very clearly to you initially, and I want to clarify how the chromosomes are. See here, this chromatid has taken the piece of the chromatid from the shaded chromosome, that is one paternal or maternal set of chromosome. So and the next one is, next chromosome is lying very nearer to this one, and it has the chromatids like this. Okay, is completely shaded chromosome. It is the chromosomes are looking like this now. So they are just like this. Okay. So this is how the pair of chromosomes, homologous chromosomes, one is from paternal set, one is from paternal set, are like this. So in metaphase, what happens over here is, here that we come across here one aster, and here also we come across one more aster. And both of these are producing what? So these spindle fibers over here, and in metaphase 1 what happens over here is, so the spindle fiber that attaches to this centromere or the kinetochore of this chromosome. See here, they attach to the centromere or kinetochore over here, and these chromosomes are lying unevenly. They are not lying evenly, and all these chromosomes are brought to the equatorial line. They are brought to the equatorial line. For your understanding, I have drawn these diagram of the chromosomes larger enough. So these chromosomes has to be brought to the equatorial line. Right? So now what happened over here? Complete disappearance of what has taken place over here? Nuclear membrane as well as nucleolus has taken place. And what is the next step? Asters are occupying their respective positions, and they produce spindle fibers. Now, right? So spindle fibers are produced, and to these spindle fibers, the kinetochore, that is chromosomal fibers, are attached. The chromosomes are attached to the spindle fibers with the help of fibers arising from the kinetochore, right? Kinetochore. So that these chromosomes are brought to the equatorial line, so that these chromosomes are brought to the equatorial line. They are brought to the equatorial line, and that process itself is called as compression. That process itself is called as compression. But the difference that we come across in the metaphase, normal metaphase as well as this metaphase 1 is nothing but, see here, when these chromosomes are brought over here, one set of chromosome, that is paternal set of chromosome, you consider this one as paternal set and this one as pattern. So both of themselves turn like this and they are lying just parallel to this equatorial line. See diagrammatically, now all the chromosomes are lying in the cytoplasm directly. So this is one aster, this is another aster. The spindle fibers are formed like this. Some are continuous and some are discontinuous fibers that we come across. So to these spindle fibers, how the chromosomes are lying with just like this, we can observe over here. This is one chromosome and this is another chromosome. See here, this is another chromosome. They are lying just like this. So one is shaded for your understanding purpose. I am shading this one. See, the chromosomes are lying just like this. Okay, they hang to the spindle fiber, they attach to the spindle fibers, and one set of chromosomes are lying towards this pole, one set of chromosomes is lying towards this pole. Is it very clear? So, just like this, the chromosomes are brought to the equatorial line, but how they are looking to be, they form the equatorial plate. Already I have mentioned it in the previous. So mitotic cell division. And how these are forming. See here, this is the centromere of one chromosome, this is the centromere of another one, another one, another one, another one. Like this, all these chromosomes are lying this. And here also the chromosomes centromere is lying this way. So these are forming one metaphasic plate and these are forming one metaphasic plate. So totally double metaphasic plate they are going to show over here. Is that very clear? So equatorial plate or the metaphasic plate they form right here in this part. When the chromosomes are brought to the equatorial line, and that process is called as compression, and they form double metaphasic plate or equatorial plate. Double, so metaphasic plate they form over here. Okay, so this is how the chromosomes are lying over here. This is one metaphasic plate, this is another metaphasic plate. All the chromosomes are lying this way. This is one set of chromosomes, this is another set of chromosomes. Okay, so now I hope it is very clear to you. Okay, this is shading, you should not forget to make over there. Okay, we have to identify them. And the terminalization that completes over here. Terminalization that completes over here in metaphase 1. After that one, these chromosomes they start to move towards this pole and this chromosome start to move towards this pole. Right? One set of chromosomes that start to move towards this pole and this chromosome start to move this pole. And next stage itself is anaphase 1, right? So the movement of the rapid movement of these chromosomes that take place in anaphase 1, right? Already we have seen this one, but the only difference what we are going to clarify over here. So this is the fact what you should remember always. Okay, so now we shall go to the anaphase 1. In anaphase 1, what happens over here is, see here, In anaphase 1, this is the cell, this is the equatorial line, this is one ester, this is another ester. So what happens over here is the spindle fiber is like this, to which the chromosomes are attached. See here, only one set of chromosomes I am considering over here. This is one chromosome. Okay. This is another chromosome. And this chromosome is completely shaded one. Okay, like this. Two chromosomes that we come across over here. So these chromosomes which hang to the spindle fiber, they start to move apart. When they completely move apart, complete disappearance of chiasmata take place. Here in this stage, both the homologous chromosomes move apart and complete disappearance of chiasmata takes place and is called as disjunction. Very important word to remember over here for NEET examination purpose. Disjunction occurs in anaphase 1 complete, and after that one chromosomes start to move towards their respective poles. They start to move towards the respective poles. So this chromosome move towards this pole and this chromosome move towards this pole. It starts to move towards the respective pole, and these chromosomes are said to be as dyads. What they are called as? Dyads, not tetrads. Previously they were tetrads in case of even metaphase 1, and there gradual disappearance of what has taken place? Gradual disappearance of the chiasmata is taking place, and the complete disappearance of complete separation of these two chromosomes that take place in anaphase 1, and that process is called as a disjunction. After disjunction, what happens? The chromosomes become dyads, right? And this is how in anaphase so the chromosomes start to move towards their respective pole upwards. So this is the fact what you should display in the diagram, as well as you have to explain thoroughly. And after anaphase 1, so there we come across telophase 1. Telophase 1. What happens in telophase 1? In telophase 1, gradually what happens here is the respective set of chromosomes move and reach towards the poles. Already the respective chromosomal group, so that reach Now I shall draw here one chromosome. Only one chromosome I shall draw here, and one more chromosome I shall draw here. What it shows is the respective chromosomes have reached the respective poles over here. See here, this has reached this pole, and this chromosomal set that reached this pole. Okay. Respectively. So now the disappearance of what takes place here? Disappearance of spindle fibers takes place over here. Disappearance of spindle fibers that take place over here, and chromosomes start to uncoil and become elongated. The chromosomes become elongated structures, thread-like structures, not completely the chromatin material, remember. So the chromosomes become very thin and uncoil, and they become elongated structures here. And the gradual appearance of what takes place over here. So now the chromosome, it starts to uncoil and start to make a thread-like structure over here. Here also it starts to make the thread-like structure over here. The gradual appearance of the nuclear membrane takes place, as well as the nucleolus takes place. The gradual appearance of nucleolus takes place here. Nuclear membrane as well as nucleolus takes place over here in this stage. And so these are all the activities that are done in telophase 1. The chromosomes become thin, elongated structures, and the appearance of nuclear membrane takes place around this chromosome and this chromosome also, and appearance of nucleolus also takes place over here. Right? So this is about what? Telophase 1. Telophase 1 is followed by the cytokinesis. Right? Cytokinesis, it is followed. Okay. See here, before this one, before the cytokinesis, we must have the knowledge of interkinesis over here. Very important term. Remember, interkinesis. What is this interkinesis? It is the stage between two meiotic divisions. It is the stage between two meiotic divisions. Divisions means meiosis 1 as well as meiosis 2. There is a interkinesis. So this interkinesis occurs over here in between two meiotic divisions. So during which there are some biomolecules are required to synthesize sometimes. So that's why there the biosynthesis of these biochemicals that take place in interkinesis. But one thing you must remember that there is no DNA duplication or DNA synthesis that takes place over here. Why? Because interphase once occurs in meiosis, already I have mentioned. So here there is no interphase at all, right? So interkinesis during which some of the biochemicals which are deficient They are synthesized over here. They are synthesized in this stage. Okay, but there is no DNA synthesis. There is no DNA synthesis that occurs during this stage. And cytokinesis. What is the cytokinesis? Cell division itself. It occurs in some cells, but it is omitted in some other cells. Means cytokinesis doesn't occur over here completely. Okay, so it is arrested there. So that's why you must remember. Here I shall explain. Cytokinesis. It is omitted in some cells, occurs in some cells. During this cytoplasmic division occurs. Cytoplasmic division that occurs over here in cytokinesis. So equal proportion of cytoplasm, all cell organelles, everything that take place over here so as to form two haploid cells. So as a result of cytokinesis, two haploid cells are formed over here. Two haploid cells are formed over here. So how it forms, that you know, in case of animals, there one of the what occurs here? There one of the cleavage that occurs at the middle part of the cell. There is a cleavage furrow is formed over here, and this is the furrow that continues over here. So and there the deposition of all those materials that take place in case of animal cell, and in case of plant cell, the accumulation of all the spindle fibers at the central axis, everything is. So it is quite natural, as if the cytokinesis of the mitosis, right? So this is how totally two haploid cells of cells are formed over here as a result of the cytokinesis if occurred in between the meiosis, means after meiosis 1. Totally how many cells are formed? Two cells are formed. By the division of one diploid cell, two haploid cells are formed in meiosis 1. And meiosis 2 is other than it is mitosis. It is just like a mitosis, nothing difference is there. So simply there the chromosomes they separate themselves and form two equal sized cells with the same number of chromosomes in daughter cells. Meiosis 2 will discuss in the next video. Thank you.